Great. So thank you very much to the organizing committee for inviting me to speak today. It's an incredible honor to be here, particularly because this award honors my long-term friend and collaborator, Dolph Schluter. And so today, my talk will actually cover a little bit of the same ground as Dolph's, but more from the perspective of genetics. Okay, and so we've already heard this quote today, but you can't start a talk on speciation without showing a picture of Darwin. And as Professor Ellegren said at the beginning, Darwin sort of raised this quote at the beginning of his book, that speciation, the origin of species, is really this mystery of mysteries. And it has remained quite an enduring mystery in evolutionary biology. But I think we've made some good progress. Oops, I didn't touch anything. Okay, so hopefully that won't keep happening. Now, before embarking on a talk about speciation, I think it's also important to sort of define what is actually a species, and for this also remains sort of a mystery of mysteries. Many people in this room wouldn't necessarily agree, but for today's talk, I'm going to use the biological species concept as my definition of a species, which is that species are groups of actually or potentially interbreeding natural populations, which are reproductively isolated. Oh, I don't know why it's automatically advancing. Are reproductively isolated from other such groups. So the study of speciation is basically the study of reproductive isolation. And so Dobzhansky, who Dolph introduced you to already, sort of went through and categorized what are possible isolating barriers between species. And we can break these into prezygotic mechanisms, so things like differences in sexual behavior, where species are breeding, when they're breeding, or if they do breed, that the sperm and eggs don't interact properly. There's also postzygotic mechanisms, which was the focus of Dolph's talk, so we can have intrinsic hybrid sterility or hybrid inviability. So if we cross things in the lab, the hybrids are dead or sterile. Or this ecologically dependent inviability that Dolph talked about, where hybrids may be inviable, but only under natural ecological conditions. Okay, so the question that's really driven my research for a long time is trying to understand the genetic architecture of speciation. And so by studying the genetic architecture of speciation is basically the genetic architecture of reproductive isolation. And this is the genetic architecture of isolating barriers. And so using these definitions, the reason I use them is it sort of gives us an inroad into attacking this problem of what is the genetic basis of speciation. Okay, so specific questions that many of us in the field are trying to address is how many loci is speciation or the formation of species just due to a few loci or many genes sort of distribute in the genome? Do these loci have large effects? So is it a single sort of locus that can have a large effect on an isolating barrier, and this sort of causes reproductive isolation immediately? Or are there many loci that each sort of contribute just a little bit to creating new species? Are these loci sort of distributed across the genome, or are there clusters of loci? And I think we'll hear some talks later suggesting the role of some clustering of loci that are important for species formation. And then, what is the molecular identity of these loci, and what can that tell us about how species form? Now, the classical approach to do this is what we call a forward genetic approach, where we can take populations or species, and if we're able, cross them, generate hybrid individuals, and then look at the location. So use genetic mapping to ask where in the genome do we see things like male sterility mapping, or maybe there's some sort of prezygotic mechanisms. And then we can sort of look at associations between genotype and phenotype. We can look in the genome sequence and say, okay, maybe in this region where we see an association between this difference in behavior that's important for reproductive isolation, what are the genes in this region, then ultimately get down to the genes and molecular changes. And this sort of forward genetic approach has actually been used to look at the genetic architecture of isolating barriers. Particularly, I would say about maybe around 20 to 25 years ago, this is where we were as a field. So a lot of work had been done in Drosophila and a few other sort of laboratory systems like mice, and mostly looking at the genetic basis of traits like hybrid sterility and hybrid inviability. Okay, but I feel like we were maybe a little bit stuck at this point, and there's really two problems with this approach. The first is that I've told you that we're interested in reproductive isolation, and as a geneticist, what you need to do is take things and cross them and get sort of viable hybrids. And so if things are by definition reproductively isolated from each other, this sort of limits the systems in which you can do these sorts of studies. The second problem, I think, is that it's sort of still unclear whether these intrinsic barriers like hybrid sterility and inviability are important for species formation, or whether they sort of form after, these barriers sort of arise after reproductive isolation in some other form has arisen, okay? And I think a major breakthrough, both for the field of speciation and also for speciation genetics, is really this insight of Dolph's that reproductive isolation evolves between populations as a consequence of adaptation to different environments, right? And so it's really thinking about speciation in sort of a different way as organisms are adapting to two environments. Maybe reproductive isolation just sort of naturally evolves as a consequence of this adaptation. So if we sort of come back to this question about what is the genetic architecture of speciation, we can sort of reframe it to say that if we understand something about the genetic architecture of adaptation, as well as these isolating barriers, maybe that can help us sort of move forward to understand the genetic architecture of speciation. Okay, and I would say as a field, we've really moved forward because of this. So there's been a lot of work in a lot of different systems now, looking at the genetic and genomic basis of adaptation to divergent environments, and also starting to look at some of these prezygotic barriers, sexual isolation, for example. Okay. And so I'm going to tell you a little bit more about sticklebacks, which Dolph also already introduced you to. But what's really nice in sticklebacks, I think, is that we have these pairs of populations that are sort of at different sort of so-called stages of speciation. And so, for example, there's these solitary lake populations where we often see disruptive selection within, for example, a lake. Where individuals seem to be specializing maybe on different food resources. We might see weak assortative mating, but really not sort of full reproductive isolation. There's also these lake stream pairs. So here we have parapatric populations that are adapted to living in the lake or the adjacent streams. Here we see divergent selection, phenotypic differentiation, but very few sort of reproductive isolation in terms of behavioral isolation. There's no post-zygotic barriers, and these are also quite young. And then we have the benthic limnetic pairs that Dolph introduced you to, where we see really strong divergent selection for feeding on different food resources. Here we see strong mating preferences, which I'll come back to. Again, no intrinsic inviability or sterility. We think that they sort of evolved there was a phase of allopatry, and then they sort of have been together for about 15,000 years. And then there's also this Japanese species pair where we actually see very strong mating preferences. And this is one case where we see intrinsic hybrid sterility. Note that these are much older species pairs, so more about a million years. And I've actually worked sort of along this continuum. And so I'm not going to talk about it today, but I've worked on these lake stream pairs now for quite some time with Andrew Hendry and Dan Bolnick. I've done a lot of work on these Japanese species pairs in collaboration with Jun Kitano, but today I want to talk about the work that I've done together with Dolph on this benthic limnetic pair, which again, he's introduced you to already. But as Dolph said, we have these repeated evolution of at least 5 independent cases of these benthic limnetic species pairs. And again, the benthic form is this large, deep-bodied form. It's adapted to eating macroinvertebrates in nearshore environments versus the limnetic form, which is found in the same lake, but is living in the open water and is adapted to feeding on zooplankton. We know a lot, actually, about the isolating barriers present between these benthic limnetic pairs. And this is work that was started by Don McPhail, who was a professor at UBC, and Dolph and many colleagues. And so we know, for example, that there is adaptation to divergent trophic niches. This is a strong driver of these benthic limnetic pairs. We also know that there's sexual isolation, so there's very strong female mate choice. So female benthics prefer to mate with male benthics, and female limnetics prefer to mate with male limnetics. We also know there's habitat isolation. So male benthics are nesting in different places in the lake than male limnetics. And again, we also know that there's a strong sort of ecologically dependent hybrid inviability, but there's no intrinsic sterility or inviability within the lab. Okay, so many years ago now, Dolph and I, along with Matt Arnegaard and Gina Conti, set out to characterize the genetic architecture of speciation in these benthic limnetic species pairs. And we focused on 2 lakes, Paxton Lake and Priest Lake. I'll mostly talk about our work today on Paxton. Again, this work was really made possible by the experimental pond facility at UBC, and also when I was in Seattle, so it was just a quick drive. Dolph still hasn't forgiven me, I think, for moving to Switzerland, but this really facilitated this really great collaboration. Okay, and again, Dolph has shown you a picture of these ponds, which again have this deep open water zone, which allows for vertical migration of zooplankton, and then this shallow littoral zone, which sort of contains macroinvertebrates. So we really do have these 2 environments present within these ponds. And indeed, you can see, for example, here's a male benthic nesting in these beds of Chara within the ponds. And here is a limnetic male who's nesting here in this more open habitat. This is his nest opening here. So these ponds really nicely replicate, I think, sort of the trophic habitat and also the habitats in which these fish are mating in the wild. And so our approach was this QTL mapping approach, again, where we crossed benthic and limnetic fish. We made F1 hybrids, and then we intercrossed those F1s to make F2s, which sort of shuffle the genotypes and the phenotypes. And then we put those F2s in the ponds, and then we measured all of these traits that we think contribute to reproductive isolation between the benthics and limnetics. We then genotyped these fish with a panel of SNP markers. This was done so long ago that next-gen sequencing was not really a thing yet. And so we had this nice panel of SNP markers developed by David Kingsley's group at Stanford. And then we do this QTL analyses where we basically ask at every region in the genome where we have a genetic marker, what's the association between genotype and phenotype? So morphological trait or female mate choice, basically the approach is the same. So in this case, the study I'm going to tell you about, we had 633 Paxton individuals, and we measured trophic niche, morphology, and hybrid performance. And we had about 400 SNP markers. Okay, and so again, this is work that Dolph sort of talked about, but I'm going to go into a few more details about the genetic component of our findings. Okay, so here is is what Dolph showed you already. So here are all 633 individuals in isotope space. So again, limnetics have this higher level of nitrogen in the wild. So we see a group of fish here that are most limnetic-like. They have high nitrogen and low carbon. And then we see this group of F2s down here, which are more benthic-like, lower nitrogen, higher carbon, and then this sort of set of intermediate individuals here. And this also maps body size, right? So individuals that are feeding on more limnetic-like resources tend to be larger, and individuals that are feeding on more benthic-like resources tend to be larger than individuals that are sort of more intermediate in their trophic niche. And so here we think that body size is really a measure of performance. So the fish that are eating well are growing well. And we can see this even more if we look at sort of what's in the stomachs of these fish. So for example, fish in this B section over here tend to be eating more of these chironomids, which are a typical benthic resource, than either the L or these very small A fish. Fish that are here in this L component portion tend to be eating copepods, which again is what they would be eating, limnetic fish would be eating in the wild. Dolph talked to you about these A fish, which are eating these terrestrial mites, which is not a normal food resource for stickleback. And then sort of everyone is eating some cladocerans. Okay, so this PC1, this major axis of variation along here, is basically what we call niche score. It's really describing the sort of benthic versus limnetic trophic niche axis. Oops. Okay, so then we wanted to look at what were the morphological components associated with this. So what do fish look like that are in the A or in the L portion of the pond or the B portion of the niche axis? And so, for example, here we see that the L fish have fewer gill rakers. So gill rakers are these structures found in the fish's mouths which allow them to process food. And so fish that are eating more limnetic prey tend to have more gill rakers because it helps them to capture these smaller prey. And so indeed, these L fish have more gill rakers. The fish that are in the B region have more this suction feeding index. So as Dolph told you, their benthics are really sucking food out of the substrate. And so basically we see this match between what the fish are eating and what their morphology is. And remember, these are F2s, so they're sort of mixing up these different components of morphology. And this really allows us to ask, what are the traits that are sort of associated with being able to feed well on a benthic resource or a limnetic resource? Okay. So, but I sort of have highlighted these traits here, but it's not that simple. So a lot of traits are sort of associated with variation in niche score. So it's really, being a good benthic fish is not just one or a few traits, it's many, many traits. And therefore, not surprisingly, what we're calling these component traits that are associated with being a good benthic or limnetic in this pond experiment map to many genomic regions. So this is just showing different stickleback chromosomes, and the different colored lines are these QTL, these regions of association between genotype and specific phenotypes. And we basically see that there's 14 of the 21 stickleback chromosomes are associated with these morphological traits. But there are some clusters. So there's this cluster here on chromosome 4, this cluster here on chromosome 16, and this cluster here on chromosome 21 that seem to be associated. Okay, so so basically we see that there's, again, this additive genetic basis for sort of position along this niche axis. So it's a complex genetic architecture. It's not a simple genetic architecture. But there are 11 genomic regions that really contribute to variation in your niche score along PC1. So this is, again, what Dolph had showed, that if we just take these 11 loci and ask how many benthic alleles does an individual have at those loci, individuals that have more benthic alleles are more benthic-like in their niche than individuals that have very few benthic alleles. So it's basically just adding up over the genome, how many benthic-like alleles do you have, and that sort of makes you a good benthic or a good limnetic. Okay. So again, I'm just going to briefly allude to these fish along PC2, these small A fish that Dolph told you about. So here is where we see this phenotypic mismatch. And so, for example, fish in this A group have a very limnetic-like opening jaw inlever and a very benthic-like upper jaw, benthic-like upper jaw protrusion. And so they basically can't feed well on anything but these terrestrial mites, which were sort of floating into the pond. And this is just to diagram these traits. Okay, and so basically, though, we could look a little bit at the genetic basis of this. So there was a QTL for opening or lower jaw inlever on linkage group 4, and a QTL for this upper jaw protrusion on linkage group 9. So they're on different chromosomes. And we actually looked at the interaction between these loci. And we found some sort of suggestive evidence for an epistatic interaction. So very similar to these Bateson, Dobzhansky-Muller incompatibilities, where you see these negative epistatic interactions, we see some supporting evidence for that if we look at these traits. But again, it's probably much more complicated than just these couple of traits and just these two loci. And again, we see in this work of Ken Thompson's, we see evidence for genome-wide intrinsic incompatibilities as well, in that fish in this particular experiment raised in the ponds have this excess heterozygosity versus fish just raised in the aquaria. Okay, so now I want to talk a little bit about this experiment we did looking at, sorry, I had a last-minute problem with my slides. The Mac to PC conversion, and so the numbers here are wrong. I had to fix all my slides. Anyways, we're going to talk about female mate choice. And so this project, so Dolph likes to give all his projects names, and this project, the female mate choice, was called Mission Impossible, because we thought it'd be so cool if we could map the genetic basis of female mate choice. But this is really hard to do. So in a traditional way, what you would do is you would have some sort of laboratory setup. You would have benthic and limnetic males that were nesting in tanks. You would put individual F2 females in there. You would see who she mates with. It'd be a ton of work to do this on the number of individuals that we needed to do for genetic mapping experiments, which requires hundreds of individuals. And so I really have to give credit to Matt Arnegard here, because he came up with, I think, a really nice design. And this is what we decided to do. We decided to use the ponds. So we set up ponds that had wild benthic or wild limnetic males. So within a pond, there was both benthic and limnetic males from the wild. And over the course of the entire experiment, it was done over several ponds, but we had about 768 F2 females. And then people snorkeled around and snoobed around in the ponds, and they would locate males' nests. So the males are territorial, they're defending their nests, and you can sort of see the males on the nest. So you pretty much know he's probably the dad of the eggs that are in that nest. And then that poor male, we took his nest away from him and gave it to a helpful helper here on the side, who then collected those nests and the eggs, put them into ethanol. And then we genotyped one egg from each clutch, plus all of the males and all of the F2 females that went in with microsatellite markers. And this allowed us to say for every egg, who was your mom and who was your dad. And then we had the information for each dad about which nest, where he was in the pond. Was he in a more benthic habitat? Was he in more limnetic habitat? And also what his phenotype was. And in the end, we had 467 unique mating events from 255 F2 females. And emphasizing again, this was a massive amount of work over a couple of seasons. So what did we find? What we found first is that F2 females, remember these are sort of a mixture of benthic and limnetic genomes, tended to mate with males that looked like them. So females that were F2 females that were smaller, so centroid size is just a measure of body size, tended to mate with limnetic males who are smaller than benthic males, and larger F2 females tended to mate with benthic males who are naturally larger than limnetics. Similarly, and I think a really cool finding here is that we had PC2 was basically a principal component axis of body shape, and we found that females mated differently depending upon their own shape. So females that were more limnetic in shape tended to mate with limnetic males, and females that were more benthic in shape tended to mate with benthic males. We also then mapped these different traits. And so remarkably, we actually found a QTL. So we just did a female mate with a limnetic or a benthic male. And this is the Mission Impossible component of it. We found a significant association here on chromosome 14 that explains some of the variation, not a lot, but some of the variation in whether females chose to mate limnetic or benthic males. We also could map this discriminant function, which sort of describes the major axis of shape variation between females. And then we also mapped size here. And so we see sort of a QTL on different chromosomes, but actually remarkably, we see this overlapping QTL between on chromosome 14. So females, both a QTL here that describes variation in shape, also describes or also contributes to variation in who a female actually mates with. Again, consistent with this sort of correlation between female shape and her mating preferences. We sort of expanded this and we went back to this model from the Arnegard study, and we used this model for niche use. So again, these 11 loci across the genome describe variation in where an individual is in sort of ecological space. And if we take the same genetic model and ask, how does it contribute to female mate choice? We see that it contributes to 21% of the variance in whether a female mates with a limnetic or a benthic male. And again, not surprisingly, because this is so strongly correlated with her shape, it also explains variation in female shape. And so what this means actually is that Dolph was right. So what we're seeing in this experiment is that adaptation to these divergent trophic niches, so whether you're sort of a benthic adapted fish or a limnetic adapted fish, that shape that an individual has somehow sort of contributes to her choice of mating and contributes to this assortative mating. And I think this is a really, really cool result. Maybe not surprising, but for me, it was just really cool from the genetic data and making these F2s to really see that we sort of see assortative mating emerge as a property of an individual sort of adaptation to these different environments. So just some answers from this work. So how many loci do we see contribute to speciation? In this case, many. And again, this sort of surprised me because these are such young species pairs. I really thought that we would see a very simple genetic architecture for these reproductive, these traits. But we don't. We see that there's many loci, that they have relatively small effects, and that they're pretty much distributed across the genome. I went into this study really thinking we were going to find just a few things, clusters of loci that may do a good benthic or a good limnetic, but we really see this distributed genetic architecture. We still have no idea what any of these genes are, and that's probably will remain a mystery for a long time because these are loci of relatively small effect, and it's hard to get down to the actual molecular variants that underlie them. So what have we learned? I think we've learned that this really distributed genetic architecture can contribute to reproductive isolation, even in the presence of gene flow. And this could be because it could be maintained because selection is so strong. Divergent selection is so strong that it sort of can maintain reproductive isolation. As Dolph pointed out, and as we'll hear from Andrew McCall in the next talk, there's probably, these variants are not new. So this is selection on ancient standing variation, and this probably facilitates this rapid evolution of reproductive isolation. And people had sort of speculated these so-called magic traits that were important for divergence to adaptation to different environments that also contributed to assortative mating, don't have to have a simple genetic architecture. And again, these genetic results are really mirrored when we do comparisons of the benthic and limnetic genomes, and Andrew's going to talk much more about that in the next talk. And then I think what's cool also is these similarities between extrinsic and intrinsic postzygotic isolation. So this sort of phenotypic mismatch and the underlying sort of genotypic mismatch that we see in so-called Dobzhansky-Muller incompatibilities that underlie things like intrinsic hybrid inviability and sterility. So with that, I want to just thank all the people who've contributed to this work. And I especially just want to thank Dolph, who has been just my collaborator, mentor, and friend for 25 years. And I've just been so fortunate to work with him. And I'm so, so pleased that he's being honored today. And I just wanted to say what I said at the speciation meeting where we learned that Dolph received this award, which was super appropriate, as that we'll talk today about scientific contributions, but I think what's also important to acknowledge is that he is a kind and generous human being who I respect very, very much. And so it's really appropriate that he received this award as well. So thank you very much.